Food is one of the basic requirements of all living organisms. The major components of our food are carbohydrates, proteins, and fats. Vitamins and minerals are also required in small quantities. Food provides energy and organic materials for growth and repair of tissues. The water we take in plays an important role in metabolic processes and also prevents dehydration of the body. Biomacromolecules in food cannot be utilized by our body in their original form. They have to be broken down and converted into simple substances in the digestive system. This process of conversion of complex food substances to simple absorbable forms is called digestion and is carried out by our digestive system by mechanical and biochemical methods. General organization of the human digestive system is shown in Figure 16.1. Digestive system. The human digestive system consists of the alimentary canal and the associated glands. Alimentary canal. The alimentary canal begins with an anterior opening, the mouth, and it opens out posteriorly through the anus. The mouth leads to the buccal cavity or oral cavity. The oral cavity has a number of teeth and a muscular tongue. Each tooth is embedded in a socket of jaw bone, Figure 16.2. This type of attachment is called thecodont. Majority of mammals including human being forms two sets of teeth during their life, a set of temporary milk or deciduous teeth replaced by a set of permanent or adult teeth. This type of dentition is called diffeodont. An adult human has 32 permanent teeth which are of 4 different types called heterodont dentition, namely, incisors, I, canine, C, premolars, PM, and molars, M. Arrangement of teeth in each half of the upper and lower jaw in the order I, C, PM, M is represented by a dental formula which in human is 2123 by 2123. The hard chewing surface of the teeth, made up of enamel, helps in the mastication of food. The tongue is a freely movable muscular organ attached to the floor of the oral cavity by the frenulum. The upper surface of the tongue has small projections called papillae, some of which bear taste buds. The oral cavity leads into a short pharynx which serves as a common passage for food and air. The esophagus and the trachea, windpipe, open into the pharynx. A cartilaginous flap called epiglottis prevents the entry of food into the glottis, opening of the windpipe, during swallowing. The esophagus is a thin, long tube which extends posteriorly passing through the neck, thorax and diaphragm and leads to a J-shaped bag-like structure called stomach. A muscular sphincter called gastroesophageal regulates the opening of esophagus into the stomach. The stomach, located in the upper left portion of the abdominal cavity, has 4 major parts: a cardiac portion into which the esophagus opens, a fundic region, body, main central region, and a pyloric portion which opens into the first part of small intestine, Figure 16.3. Small intestine is distinguishable into 3 regions: a C-shaped duodenum, a long coiled middle portion jejunum, and a highly coiled ileum. The opening of the stomach into the duodenum is guarded by the pyloric sphincter. Ileum opens into the large intestine. It consists of cecum, colon, and rectum. Cecum is a small blind sac which hosts some symbiotic microorganisms. A narrow finger-like tubular projection, the vermiform appendix, which is a vestigial organ, arises from the cecum. The cecum opens into the colon. The colon is divided into 4 parts: an ascending, a transverse, descending part and a sigmoid colon. The descending part opens into the rectum which opens out through the anus. The wall of alimentary canal from esophagus to rectum possesses 4 layers, Figure 16.4, namely serosa, muscularis, submucosa and mucosa. Serosa is the outermost layer and is made up of a thin mesothelium called epithelium of visceral organs, with some connective tissues. Muscularis is formed by smooth muscles usually arranged into an inner circular and an outer longitudinal layer. An oblique muscle layer may be present in some regions. The submucosal layer is formed of loose connective tissues containing nerves, blood and lymph vessels. In duodenum, glands are also present in submucosa. The innermost layer lining the lumen of the alimentary canal is the mucosa. This layer forms irregular folds, rugae, in the stomach and small finger-like foldings called villi in the small intestine, figure 16.5. The cells lining the villi produce numerous microscopic projections called microvilli, giving a brush border appearance. These modifications increase the surface area enormously. Villi are supplied with a network of capillaries and a large lymph vessel called the lacteal. Mucosal epithelium has goblet cells which secrete mucus that help in lubrication. Mucosa also forms glands in the stomach, gastric glands and crypts in between the bases of villi in the intestine, crypts of Lieberkühn. All the four layers show modifications in different parts of the alimentary canal. Digestive glands. The digestive glands associated with the alimentary canal include the salivary glands, the liver and the pancreas. Saliva is mainly produced by three pairs of salivary glands, the parotids, cheek, the submaxillary/submandibular, lower jaw, and the sublinguals, below the tongue. These glands situated just outside the buccal cavity secrete salivary juice into the buccal cavity. Liver is the largest gland of the body, weight about 1.2 to 1.5 kilograms in an adult human. It is situated in the abdominal cavity, just below the diaphragm, and has two lobes. The hepatic lobules are the structural and functional units of liver, containing hepatic cells arranged in the form of cords. Each lobule is covered by a thin connective tissue sheath called the Glisson's capsule. The bile secreted by the hepatic cells passes through the hepatic ducts and is stored and concentrated in a thin muscular sac called the gallbladder. The duct of gallbladder, cystic duct, along with the hepatic duct from the liver forms the common bile duct, figure 16.6. The bile duct and the pancreatic duct open together into the duodenum as the common hepatopancreatic duct, which is guarded by a sphincter called the sphincter of Oddi. The pancreas is a compound, both exocrine and endocrine, elongated organ situated between the limbs of the C-shaped duodenum. The exocrine portion secretes an alkaline pancreatic juice containing enzymes, and the endocrine portion secretes hormones, insulin and glucagon. Figure 16.6. The duct systems of liver, gallbladder and pancreas. Digestion of food. The process of digestion is accomplished by mechanical and chemical processes. The buccal cavity performs two major functions: mastication of food and facilitation of swallowing. The teeth and the tongue with the help of saliva masticate and mix up the food thoroughly. Mucus in saliva helps in lubricating and adhering the masticated food particles into a bolus. The bolus is then conveyed into the pharynx and then into the esophagus by swallowing or deglutition. The bolus further passes down through the esophagus by successive waves of muscular contractions called peristalsis. The gastroesophageal sphincter controls the passage of food into the stomach. The saliva secreted into the oral cavity contains electrolytes and enzymes, salivary amylase and lysozyme. The chemical process of digestion is initiated in the oral cavity by the hydrolytic action of the carbohydrate-splitting enzyme, the salivary amylase. About 30% of starch is hydrolyzed here by this enzyme, optimum pH 6.8, into a disaccharide, maltose. Lysozyme present in saliva acts as an antibacterial agent that prevents infections. Starch turns into maltose by salivary amylase in presence of 6.8 pH. The mucosa of stomach has gastric glands. Gastric glands have three major types of cells, namely I. Mucus neck cells which secrete mucus. 2. Peptic or chief cells which secrete the proenzyme pepsinogen, and 3. Parietal or oxyntic cells which secrete HCl, an intrinsic factor essential for absorption of vitamin B12. The stomach stores the food for 4 to 5 hours. The food mixes thoroughly with the acidic gastric juice of the stomach by the churning movements of its muscular wall and is called the chyme. The proenzyme pepsinogen, on exposure to hydrochloric acid, gets converted into the active enzyme pepsin, the proteolytic enzyme of the stomach. Pepsin converts proteins into proteoses and peptones or peptides. The mucus and bicarbonates present in the gastric juice play an important role in lubrication and protection of the mucosal epithelium from excoriation by the highly concentrated hydrochloric acid. HCl provides the acidic pH, pH 1.8, optimal for pepsins. Renin is a proteolytic enzyme found in gastric juice of infants which helps in the digestion of milk proteins. Small amounts of lipases are also secreted by gastric glands. Various types of movements are generated by the muscularis layer of the small intestine. These movements help in a thorough mixing up of the food with various secretions in the intestine and thereby facilitate digestion. The bile, pancreatic juice and the intestinal juice are the secretions released into the small intestine. Pancreatic juice and bile are released through the hepatopancreatic duct. The pancreatic juice contains inactive enzymes, trypsinogen, chymotrypsinogen, procarboxyptadases, amylases, lipases, and nucleases. Trypsinogen is activated by an enzyme, enterokinase, secreted by the intestinal mucosa into active trypsin, which in turn activates the other enzymes in the pancreatic juice. The bile released into the duodenum contains bile pigments, bilirubin and biliverdin, bile salts, cholesterol and phospholipids but no enzymes. Bile helps in emulsification of fats, that is breaking down of the fats into very small micelles. Bile also activates lipases. The intestinal mucosal epithelium has goblet cells which secrete mucus. The secretions of the brush border cells of the mucosa along with the secretions of the goblet cells constitute the intestinal juice or succus entericus. This juice contains a variety of enzymes like disaccharidase, example maltase, dipeptidase, lipase, nucleosidase, etc. The mucus along with the bicarbonates from the pancreas protects the intestinal mucosa from acid as well as provide an alkaline medium, pH 7.8, for enzymatic activities. Submucosal glands, Brenner's glands, also help in this. For part 2, press the link right on the corner.